Explain everything. Cellular respiration. Cellular respiration is a 4-step process that starts with glycolysis. Next is pyruvate oxidation, followed by Krebs cycle, and then electron transport chain. To start cellular respiration, we need oxygen and sugar. What we get out of it is water and CO2. This is the balanced chemical equation. The first step of cellular respiration, glycolysis, is the breaking down of a glucose molecule into 2 pyruvate molecules. This is performed in the cytoplasm. We start off with 1 glucose at the top here. The first step in glycolysis is energy requiring, which means we need 2 ATP molecules to be hydrolyzed. So ATP is transformed into ADP here. This attaches a phosphate group to the glucose, making glucose 6-phosphate, or G6P. Glucose 6-phosphate is then rearranged into fructose 6-phosphate. Another ATP is used and changes to ADP here, and a phosphate group is added to the fructose 6-phosphate, creating fructose 1,6-biphosphate. Fructose 1,6-biphosphate is then split, making DHAP, right here, and G3P, after which DHAP is then turned into G3P as well. So this would continue down this direction, but we're just going to have it set up like this for now. G3P then becomes oxidized using NAD+, here, which becomes NADH, which becomes important later on. This oxidation reaction also releases energy, so this allows BPG to turn into 3PG. It also turns another ADP to ATP, which is what we want. Next, 3PG is rearranged into 2PG. At this point, 2PG is converted to PEP by removing a water molecule here. The phosphate group on PEP phosphorylates an ADP into an ATP, changing PEP into pyruvate. That's the end of glycolysis. The next step in cellular respiration is pyruvate oxidation. This occurs in the mitochondria. Pyruvate molecules enter the matrix, which is the inside of the mitochondria, via the help of membrane carriers. The pyruvate molecules cross the membrane, and coenzyme A attaches to the acetyl group of the pyruvate molecules, which enables it to be oxidized. NAD+ is reduced and becomes NADH. The remaining carboxyl group is removed as waste as CO2 here, and the resulting product is acetyl-CoA, which remains in the matrix. When energy is needed, acetyl-CoA enters the Krebs cycle to continue aerobic respiration. This is the next step of cellular respiration. Krebs cycle is the next stage in cellular respiration. It's a series of 8 reactions in order to transfer energy from organic molecules into usable forms for the cell, such as ATP, NADH, and FADH2. It also releases CO2 as a waste product. For every glucose molecule that enters glycolysis, 2 cycles of Krebs occur. This is when it's split into 2 on the glycolysis slide. Krebs cycle occurs in the matrix of the mitochondrion. and each of the 8 reactions is catalyzed by a specific enzyme. Step 1 begins with acetyl-CoA from a pyruvate oxidation, which was the last thing we did, which reacts with oxaloacetate, which is in the matrix already. This forms citrate. Next, citrate undergoes an isomerization to form isocitrate. Isocitrate is oxidized and becomes alpha-ketoglutarate. A NAD becomes NADH at this point as well. Alpha-ketoglutarate makes succinyl-CoA after binding with a CoA. It also is oxidized by NAD+, which becomes NADH. CO2 is also released at this point. Succinyl-CoA loses its CoA after a phosphate displaces it. Guanosine diphosphate is phosphorylated and becomes GTP. Then the phosphate is transferred from ADP and creates a new ATP. Here. FAD comes in from the matrix and oxidizes succinate. FAD becomes FADH2 and succinate becomes fumarate. Next, water is added to fumarate, which makes malate. Malate is oxidized by NAD+, which becomes NADH, and malate becomes oxaloacetate. And thus the Krebs cycle repeats. Pyruvate oxidation produces 2 molecules of NADH, and Krebs cycle produces 6 molecules of NADH, 2 ATP, and 2 FADH2. This is for each molecule of glucose that enters cellular respiration. Electron transport chain takes place in the inner mitochondrial membrane. ETC makes ATP through oxidative phosphorylation. There are 7 major components of the electron transport chain, which are all found in the inner mitochondrial membrane. Complex 1, here, is NADH dehydrogenase. Complex 2, here, is succinate dehydrogenase. Complex 3 is cytochrome BC2 complex. And complex 4 is cytochrome oxidase. The 2 electron carriers are UQ here and site C here. The proteins in the ETC are set up in order of increasing electronegativity, with NADH dehydrogenase being the lowest. That makes the first component the weakest electron acceptor and the last component the strongest electron acceptor. Electrons are passed from one component to the next along the membrane. NADH, over here, binds to NADH dehydrogenase, which is the first component. FADH2 binds to succinate dehydrogenase, which is the second component. These hydrogen ions are then pumped into the intermembrane space. The mobile electron carrier, UQ, carries electrons for NADH dehydrogenase to cytochrome BC1. Site C then carries electrons from complex 3 to complex 4. FADH2 works the same, but instead it starts at succinate dehydrogenase. Since electrons from FADH2 pass through fewer electron carriers, it makes less ATP. Each molecule of NADH produces 3 ATP and 3 hydrogen ions. Each molecule of FADH2 makes only 2 ATP and 2 hydrogen ions. When the 2 electrons leave cytochrome oxidase, they combine with oxygen, the final electron acceptor, and H positive to form H2O, which comes out here. oxygen is used because it is the only element with a strong enough electronegativity to strip the last component of the ETC of its electrons. ETC creates an electrochemical gradient to power ATP synthesis. Chemiosmosis is possible only after the electrochemical gradient has been created by the ETC. Those hydrogen ions are now forced through a special enzyme called ATP synthase, which is embedded in the inner membrane. Hydrogen ions then flow from the inner membrane space into the matrix. The flow from hydrogen ions provides energy to make a bond between ADP and inorganic phosphate. This is how ATP is made via oxidative phosphorylation, indirect formation of ATP through oxidation.